Hey guys, welcome to chapter 6. We are starting photosynthesis this chapter. It's a concept you've probably seen, most certainly have seen before in biology, but we are going to dive quite a bit deeper into a lot more detail than we have previously with photosynthesis. And so this is one that's going to require some studying. It's going to require, some flashcard making or something to kind of get through these biochemical pathways because they are a little tricky and they're tough to remember. So I'm going to give you guys just every tool I can to kind of help you learn these, and we'll do some videos and things like that that'll help us get through these. So where it starts, photosynthesis. We got energy coming from the sun, and it's not very useful for you and I, but thanks to plants, they're going to make it useful, and they're going to do so in a hurry. So, your book starts with a brief introduction on biofuels. You may have heard of these, you may have not. Biofuels is the idea of making a fuel out of something that is living. We start with autotroph, an idea that you are already familiar with. Autotrophs, they make their own food. We call these guys the self-feeder. They're going to harvest energy from their environment, normally the sun, but not necessarily. There are a few bacteria that are chemoautotrophic and can actually just eat chemicals. So that's kind of cool. But most of these guys are using the sun, and they can get carbon from their inorganic molecules. And so that's pretty cool. So photosynthesis gets the energy from the sunlight. We see this primarily in plants. Most of our autotrophs, bacteria, algae, those kind of guys, are using photosynthesis to make their organic molecules, primarily glucose, and that we're going to use as food, also as building materials too. So that's something to keep in mind. Pardon me. So heterotrophs, that's you and I, we get our carbon from the organic molecules made by the autotrophs. So the plants make the sugars, we eat the sugars, even though it's not sweet like sugar necessarily from sugar cane. When you're eating vegetation, you are eating the sugars that they've produced from the sun. Today we use a lot of fossil fuels, and we have for a while. Fossil fuels are how we're getting things like our methane, our propane, our gasoline, our diesel, those kind of things that are running cars and heating homes and such. These are the remains of ancient forests that have been kind of compressed, and we get this really dense source of carbon that we can burn, and that's what we use for all of these things. The bad thing about fossil fuels is they are a limited resource. There are a lot of disagreements on where those limits are, how long they can sustain us, but one thing is certain, we will eventually run out of fossil fuels. It's gonna happen. And when it does, we need to be ready with other energy sources, because none of us wants to stop driving our cars and start walking to work every day, especially since our school is so far away from your home. So biofuels are made from organic matter that is not fossilized, and so that makes them a renewable resource, because fossilization is not happening to really any extent that we're aware of currently today. So we need a renewable resource that doesn't require fossils, and that would be corn, soybeans, sugarcane, switchgrass, that kind of stuff. So the idea is, we grow the crop, we cut it down, we make the fuel. It's just like fueling our bodies. We grow the crop, we cut it down, we eat the food, and we fuel our bodies. Well, now we're going to take some land and we're going to grow the crop, harvest it, make fuel, put the fuel in our cars. And you've already seen this with ethanol introduced into a lot of gasolines already here in the US. So this isn't totally new to you, but we're looking for crops other than corn, because corn can also be used as a food source. Whereas something like a grass, for example, a switchgrass or something like that, maybe we could get a lot of yield out of that. And you and I normally don't sit down to eat very much grass. So that would be one option we could approach. Researchers are looking for ways to use our non-food plant matter, like switchgrass or agricultural wastes, wood chips, that kind of stuff, to make a fuel out of those. And so that's an important application of biology and chemistry together. So sunlight is the energy source, Energy flows through all our ecosystems, and it begins when our photosynthesizers, those are plants and algae, intercept energy from the sun. And the way they do it is they use what's called a pigment. A pigment is a pretty long molecule that captures sunlight energy and converts it into chemical energy, which when you think about it is an incredible feat. And really, so much sunlight comes to the earth, and it hits you and I, and we get like sunburns, and it hits the dirt, and nothing happens, and all that energy is just wasted. And it's really the plants and the bacteria, the photosynthetic bacteria and photosynthetic protists, that are able to take something, pardon me, that's not particularly useful and make it very, very useful and provide a foundation for entire ecosystems and entire biospheres to grow from. So that's pretty incredible. And they're going to store that energy in chemical bonds, and once it's in a chemical bond, it is accessible to essentially everyone. So, properties of light. We're going to look at this sunlight stuff that's pouring down to Earth for a lot of the day. Visible light is part of a much larger electromagnetic spectrum of energy, and all of this stuff is radiating from the sun. It travels in waves, and the visible light is organized into photons. Photons are like a packet of solar energy. So when we're talking about waves, we can talk about things like X-rays, things like infrared, gamma rays, all sorts of stuff. We're going to keep it pretty narrow in biology to just worry about the light waves that are responsible for photosynthesis and responsible for our vision, and that's about where our concern about ends. But wavelength is the distance between the crests of two successive waves of light, and that's very important. So if I take my really poor drawing skills on my computer and I draw some waves, this is, I don't know, red, why not? It's a wavelength of red light. Okay, there's a few waves, and it repeats and repeats and repeats and repeats. The wavelength is this distance from crest to crest. That is one wavelength. And we could measure that and say, well, that is, I don't know, 700 nanometers or something like that. And that is a measurement of wavelength. And that tells us a lot about this wave. If you wanted to go trough to trough, you could. Crest is the top, trough is the bottom. So there's trough, and you would measure that. Again, you would get 750 nanometers. Even with my poor drawing, you can see that they're approximately, very close together. And you would get the same wavelength. So the shorter your wavelength, the greater your energy. So red's pretty long, but if we were to look at something like, I don't know, blue, it might look more like this. Okay, bad drawing, I know, I'm sorry. There you go, okay? So its wavelength, if you were to measure crest to crest, would be very, very short. And all that really means is that this guy up here is carrying a lot more energy than this guy down here. Okay? And we can use longer wavelengths to do things like broadcast radio signals to your cars, and we can use shorter wavelengths to like take pictures of your bones. They've all got uses. They're just very different in their properties of a wave. So, shorter wavelength, greater energy. Good? Good. Alrighty, so here's where we fit. Gamma rays make the Incredible Hulk. X-rays take pictures of your bones. UV is the stuff that's coming from the sun. When you buy sunglasses, they'll say stuff like UVA, UVB blocking, or whatever. That's the stuff that sunburns you. And then this little teeny tiny baby sliver of that whole spectrum is what you and I see. And see, they've got it going through a little prism back here. It's kind of cool. So the sunlight comes in and poof, a prism separates it out into its independent colors. And what you get is a really brief range from 400 nanometers to 700 nanometers. And that is what we call visible light. Plants are using this. You and I are seeing this. And there are a few critters out there that can kind of see a little bit in this range and kind of sense a little bit in this range. But for you and I, we're really confined to a very small portion of a much larger spectrum. So write that down, please. 400 nanometers to 700 nanometers. That is visible light. All right. You get near infrared and infrared, which is like thermal imaging camera kind of stuff, which is sort of neat. Radiation that's in the infrared, which is again thermal imaging. They have heat escaping from the Earth's surface, so that's kind of cool. The Earth, giving off heat. And then when we get way out here, there's microwaves, like what heats our food. And then way out here is radio waves, which if we wanted to, we could broadcast a very, very, very long ways, a lot further than the, 90-mile radius that we use here in Rapid. If you wanted to broadcast radio waves, across the world, basically, you could do that without too much trouble. So they've all got their uses. Biology is concerned with a very small portion of it, and physics is concerned with a much larger portion of it. So we're going to stick with the biology part. The different wavelengths of light form the colors of the rainbow. You just saw that on the last slide. Photosynthesis is going to use a wide variety of these wavelengths depending on the pigment, but it's going to range 380 to 750. So if you look back here, 380 to 750. So between friends, we can pretty much say it's between 4 and 7. I mean, if you want to be technical, it's 380 to 750, but we're pretty much between 4 and 700 nanometers. So a pigment is an organic molecule. They're normally pretty decent sized. They selectively absorb light at specific wavelengths. So you might have a pigment that absorbs light, between 380 and 430. And that's about it. If light comes in above or below that, it's going to pass right through and it's not going to bother that pigment at all. So pigments are very specific. Chlorophyll A is our most common photosynthetic pigment, and it absorbs primarily violet and it absorbs red. And so what you get with chlorophyll A is it absorbs this stuff. It absorbs this stuff. And so what it doesn't absorb, it reflects. So if the plant is absorbing purple and it's absorbing red, then it will be reflecting primarily green or yellow. And when you think about a lot of the plants that we see out in the world, we're seeing greens and yellows before we're seeing purples and reds. Not to say that there aren't plants that have purple parts or red parts, there are, but for the photosynthetic part of the plant, primarily green is what we're looking at. So that's chlorophyll A. We're absorbing purple, we're absorbing red, and so as a result, the plant is appearing green, or the bacteria appears green, or the algae appears green, etc., etc. So collectively, in any given plant, you get chlorophylls, you have an A and a B, and you also have a bunch of little accessory pigments that I don't want to say aren't that important, but they aren't that important, and they're actually going to absorb most of these wavelengths of visible light. So they're going to try to get as much as they possibly can out of that light. Certain electrons in these pigment molecules absorb photons of light energy, and it boosts those electrons to higher energy levels. So you guys learned in chemistry how electrons could be in a ground state or in an excited state. And now that we're in biology, we're learning that photons, the energy from the sun, hits pigments, transfers the energy from the sun to the electron in the pigments and excites the electron. And once that happens, we have harvested energy from sunlight. And it's really incredible, but chemistry is the foundation for what we're going to talk about when we get to photosynthesis here. So that energy is now the plant's. It has captured it. And once it has captured it, it kind of passes it around like hot potato and gets that electron bouncing all over, and everybody starts to get energy from it. And we can use that for photosynthesis. So, couple pigments. You do not have to memorize this chart, but here they are. This is chlorophyll A. It's a pretty big molecule. There's a little magnesium atom in the middle. This guy's the one who's going to get excited by electrons. These guys are going to kind of pass it around to him. It's sort of cool. So chlorophyll A is going to appear green. You're going to find him in plants. You're going to find him in protista, like euglenoids and photosynthetic protists and algae. And you're going to find him in bacteria. You're not going to find him in archaea. Okay? There are some other chlorophylls that work in a manner very similar to this, and some phycobilins that are going to appear blue, red, violet. They're going to reflect some other wavelengths, and really they're just harvesting light in a slightly different wavelength than these chlorophylls are. And so you can see we're not finding a lot of phycobilins in plants. They're pretty much in our protists and our bacteria. And so that's kind of a way you can tell a little bit about where they live. These protists and bacteria are a little deeper in the pool a little deeper in the pond, and so they're going to get some wavelengths that travel a little further than that green does, or than the red and the violet do. So we go down to carotenoids. It's where, carrots are orange, carotenoids are orange pigments. And beta carotene is one that you've heard of, I'm sure. There are a bunch of others that you don't have to remember their names, although you might recognize that one from Gen Bio. It's what makes brown algae brown. We did talk about that. And what we get is these guys make plants a little different color. So some leaves have red in them, some have yellow, some have brown, and we find these carotenoids in plants, we find them in protists, we find them in a couple of bacteria as well. And then finally, way down here, we get anthocyanins. This one's called retinal. It's purple, as you can see here, and its color is going to be violet because it's reflecting that violet. And we really only find this one in Archaea. So kind of cool. There's a lot of variety in the pigments, and they're absorbing different frequencies. I shouldn't quite say that. Wavelengths. They're absorbing different wavelengths of light. And when they all work together, you see how many of these are in plants. When a plant has multiple pigments like this, it can absorb a lot of solar energy, and not a whole lot's going to escape through. So it's really important. So photosynthetic pigments, they work together. They harvest light of their different wavelengths like we just talked about. And there was this scientist called Engelmann who did this really cool experiment where he identified the colors of light that were driving photosynthesis long before we had the ability to do this with any sort of electronic equipment or anything like that. And he used a prism, so a very cool example of what you could almost call like a low budget minimalist approach to gathering data, and he still got data from it. So I'm going to show you this video of his experiment real quick.
Engelmann placed a strand of the green alga, Spirogyra, on a microscope slide. He added water that contained oxygen-requiring bacteria. The bacteria distributed themselves randomly on the slide. Engelmann placed the slide on a microscope and illuminated it using light filtered through a prism. This breaks up light into its component wavelengths. Over time, the bacteria were attracted to areas where oxygen was being produced by photosynthesis. This was primarily where violet and red wavelengths crossed the algal strand.
So there you have it. It's a way you can gather data, the number of bacteria, the concentration of bacteria, based on the areas where you are hypothesizing that the most oxygen is going to be produced, because they're going to produce the most oxygen where they're carrying out the most efficient photosynthesis. And so in the areas where those pigments were able to absorb light, like the purple and the red, they made more oxygen. The bacteria that required oxygen slowly swam themselves over there, and you found more bacteria here. Now in the yellows and the greens, where the algae is reflecting that light, it's not making much oxygen from photosynthesis in here, and so really not many bacteria want to be in there. So we got data long before we had the really cool technology that we have today to get that kind of data. So very cool experimental design. So there's Engelmann's experiment, and here's what it looks like. This guy's Spirogyra, or Spirogyra. He gets the name because of his long spiraled chloroplast, which looks super cool. And then he has the light coming in that's irregular light from a microscope with a prism. The prism breaks it out into its colors, and you can see your bacteria. They're all about this purple area. The green, not so much. The red, pretty good. And it gives us a good idea of what wavelengths of light plants can actually use for photosynthesis. So most of our photosynthetic organisms are going to use a combination of pigments to drive photosynthesis. You're not going to find many plants that just have chlorophyll B and that's it. And that's all. No, of course, they're going to have many different pigments. And this absorption spectrum shows which wavelengths each pigment absorbs the best. So our organisms in different environments use our different pigments. And I kind of explained that to you earlier, how, well, the bacteria and the protists, they might kind of be hanging down deeper in the pool, so they're going to want a wavelength that's a little more powerful, a little shorter wavelength that's going to penetrate deeper into the water, Whereas, the leaf at the top of the tree, he has his pick of whatever light he wants, so he can take chlorophyll A and B and make energy for days. It just depends on where you live. And here they are. So light absorption, the higher your peak, the more light you are absorbing, okay? Now, the wavelength is our visible light range. You can see it does go down kind of towards that 380. We started at 400, but you'll know there's nothing, those lines would keep going. And then once you get past about that 750, there's really not much. So here's something cool to look at. I want to show you this kind of, I guess it'd be a teal line. It's chlorophyll A. Some of these pigments have multiple peaks. Okay, so chlorophyll A, if you follow that weird teal color, actually starts here. And he peaks in the kind of 450, maybe about 430 to 450 range. He peaks in that range. He drops down through our greens and yellows, and then that teal pops up around our orange and red. 600 to 700, here comes chlorophyll A. So he actually has two peaks. He works at both of those wavelengths, which correlates really well with Engelmann's experiment. Now, like I said, plants aren't alone in those peaks. So where chlorophyll A starts to dip down, green chlorophyll B starts to peak. So even though light might be coming in at more that 470 wavelength and chlorophyll A can't catch it, that's fine because chlorophyll B can catch it. He's still going to dip down, chlorophyll B, around that green and yellow wavelength, and then he's going to pop up a little bit before chlorophyll A is in about that 640 to 650 range. So those are our two heavy hitters for photosynthesis. You can see beta carotene does a lot of overlap with these guys. And then we get a couple that are in this green range that are actually going to absorb this light that's in the green and yellows. And those are phycoerythrobilin. I hope you don't have to spell that correctly because that is crazy. And phycocyanobillin. And so these are going to be a few of our bacteria and protists that really aren't getting, I guess, the pick of the litter when it comes to wavelengths that are really great for photosynthesis, but they have pigments that are specific enough that they're going to catch what energy's there and make the very best they can of it. So out of the visible wavelength, when we consider plants, bacteria, protists, and archaea, what's absorbed? Our answer is pretty much all of it. I mean, they're not going to get every photon that falls from the sun, but they're getting a lot of across the visible wavelength. They really are. So they work together. In the few photosynthetic eukaryotes that we see, because that's kind of odd, but we do have some plants that it's not odd, eukaryotes, multicellular, organelles, those kind of guys, you and I. There are many photosynthetic eukaryotes. Plants and trees, they're all eukaryotes. Photosynthesis occurs in an organelle called a chloroplast. You remember that a prokaryote did not have organelles. A eukaryote has organelles. And so we have our own organelle for photosynthesis called a chloroplast. And it's an organelle that specializes just for photosynthesis. We find it in plants. We find it in some protists like Euglena. And our photosynthesis occurs in two stages. It's the light-dependent stage and the light-independent stage. Now, those two topics are pretty detailed, and we're going to save those for another lecture. So that's where we're going to stop for today.